So
right?
oxidizing LDL is one way to modify the LDL to induce the macrophages to overeat, which is going to end up causing us problems.
Now, why could that be the case? Well, LDL has, and I'm going to spend most of my time talking about LDL here just
Mhm.
because, it is 95% and it is the main part of the story in the common model that I'm going to critique here. LDL has other roles. Okay. It's a antibiotic.
Hmm.
Okay. So when you get an infection, one of the things that the bacteria try to do is form a colony, right? Biofilms people have heard about. An LDL particle can be oxidized in the bloodstream by another type of white blood cell called a neutrophil. And an oxidized LDL particle is able to prevent these bacteria from functioning normally. It interferes with what's called their quorum sensing mechanism. And that helps prevent the body from an infection when you get bacteria into the
Mhm.
bloodstream, right?
It's messing with the ability of the bacteria to coordinate with each other, which would result in a worse infection.
So that's you know, neutrophils, these white blood cells, do what's known as an oxidative burst, where they basically use their mitochondria, ramp up their mitochondria, produce a lot of very toxic molecules, and spew those out at the bacteria, right? That's all great. So you could see then, if you have FH and you're living in, dirty medieval Europe,
Right, right, right.
having high levels of LDL could be beneficial to you. It
Wow.
could help you
Okay.
withstand infection.
Yeah.
So, we've identified two problems with the model.
Mhm. When you say the model, right, the idea here is okay, we've got these plaques, they're made out of cholesterol. If someone has high cholesterol, maybe that's making more plaque and then driving heart disease. But what you told us a few moments ago was, well, actually, in these people with genetically high levels of cholesterol, familial hypercholesterolemia, they've got a deficit in the LDL receptor. And so even though their cholesterol levels are high in the bloodstream, they actually aren't getting into
Right.
the plaques, which is the opposite of what you would expect in the sort of naive model.
And so in the effort to try and elucidate exactly what mechanism is going on, specifically in FH people, a couple of scientists prior to this paper, but discussed in this paper, Brown and Goldstein, discovered the LDL receptor. Okay. They got a Nobel Prize for doing that. And then the first thing that they did is they took LDL and they put it in a vial with, this is the in vitro study, right? They put it in a vial with macrophages and they waited for the macrophages to be converted into foam cells
Mm-hmm.
because that's the first part of atherosclerosis is the appearance of foam cells.
Yep. So the macrophages are somehow getting sick, let's say, or not quite doing what they're supposed to do,
Over-consuming.
over consuming.
The problem is it didn't work,
Hmm.
right? Macrophages with what they started calling native LDL uh, do not become foam cells, right?
Hmm.
So then they tried, I forget exactly why they did this, but they came up with what they referred to as a modified LDL. It was acetylated, right? Which is not a process that happens in the body. But it did have the effect of causing the macrophages to gorge on
Hmm.
modified LDL and become foam cells.
So this starts to tell us, okay, when the LDL is modified in certain ways, that can induce overconsumption by the macrophages.
But they didn't know what it was because they knew that the acetylated LDL that they were using doesn't appear in the body,
So there
right?
could be some other way that LDL is modified naturally in a body in vivo that would
Right.
cause the same effect.
So Whitstone and Steinberg in this other paper, they're the guys who figured out what was going on. And Brown and Goldstein wrote a very nice paper acknowledging that they had discovered it and solved the problem that they had found. And this is a later paper in a series of papers from Whitstone and Steinberg discussing what's going on, right?
Hmm.
So it turns out that the oxidized LDL that I mentioned, if you incubate macrophages with oxidized LDL, they will ingest enough of it to become foam cells,
So
right?
oxidizing LDL is one way to modify the LDL to induce the macrophages to overeat, which is going to end up causing us problems, and oxidation, as opposed to the acetylation experiment you mentioned a moment ago, this does happen inside of us.
Right. It does happen inside of us. And particularly it happens inside of the artery, although it happens in other places as well.
And I would imagine it's because, right, we're talking about blood flow through the cardiovascular system. There's lots of oxygen. So you're going to get oxidation.
So what's getting oxidized is the next question,
Right.
And the payload of a LDL particle, as I said, it's cholesterol and fats. The largest part of the fat that's in the uh, particle is a fat called linoleic acid, right? The other fats tend to be fats that your body can make. Okay. Linoleic acid is unique in the payload in that your body can't make it. It has to be consumed.
So LDL particles tend to have a disproportionately high amount of linoleic acid, i.e., they tend to have disproportionately high levels of fatty acids that your body cannot synthesize itself.
And they also have a lot of antioxidants that can, linoleic acid is very fragile fat, very susceptible to oxidative damage, which is, the oxidation in the oxidized LDL. It's oxidized linoleic acid almost.
And this tells us something. Whenever you have something that's easily oxidizable, like linoleic acid in nature, whenever you look at where this stuff shows up, whether it's in a seed or in a fish or in what Tucker's talking about right now, when biology is doing it on purpose, so to speak, the easily oxidizable polyunsaturated fatty acid is almost always packaged with antioxidants.
Right.
And the way you interpret that is the biology is trying to prevent oxidation of this thing.
Well, it's trying to control it.
Contain it, regulate it.
Because, one of the other fats that's in, we get, again, this distinction between animal lipids and plant lipids. One of the other fats that's, found in the LDL particle is arachidonic acid, which is another polyunsaturated fat from the omega-6 family. And that comes from animals, right? Linoleic acid, the primary source of that in our diet is from plants. Although if you eat a cow, you will get some amount of linoleic.
Okay. So you can modify LDL particles and the macrophages will then overeat and this will end up causing problems. One way to modify the LDLs to induce that effect is to oxidize them. Oxidation is happening in our bodies all the time because it's an oxygen-rich environment, and linoleic acid is present at particularly high levels in these LDL particles.
So Steinberg and Whitstone in this 1991 paper conclude the nature of the substrate for lipid peroxidation, meaning the oxidation that's going on in the LDL, mainly the polyunsaturated fatty acids in lipid esters and cholesterol, is a dominant influence in determining susceptibility to atherosclerosis. As noted by Esterbauer, there's a vast excess of polyunsaturated fatty acids in LDL in relationship to the content of natural endogenous antioxidants, right? So
you've got more polyunsaturated fatty acids in these particles than is than the antioxidants can handle. So then they're easily oxidized. And then that is what's going to cause the macrophage overeating problem.